So, good evening, everyone. I am Vedika, and I've been a research fellow with OmicsLogic from the last month of September. And I got introduced to this platform as we had to do a minor project in college, and we were just looking for internships. Most of the tutorials were closed because of the coronavirus. So, my juniors introduced me to this tutorial, and I thought, yeah, it would be a great opportunity to learn bioinformatics, as you know, it's the need of the hour, and most of the drug designing or looking at the Are you able to hear me?
Yes,
Somebody, I
very
just
clearly.
okay, having trouble hearing
Yes,
me.
yes.
Okay, so this is how I got started. That we had to do this internship, and I mean my minor project. So I came here, and I'll talk a little bit about me. I have a basic degree in biotechnology. I've also done my master's in biotechnology. And can we have the next slide, please? So my research interests are, I'm very interested in neurobiology, especially being able to look at the gut microbiota and its relationship with diseases and the development of the brain. Then I also got interested in cancer transcriptomics when I came here, and virology. These are my basic three interests. So interestingly, before I came here, I just knew enough bioinformatics for me to write my midterm exams and assignments, and I can navigate NCBI. That was the kind of person I was. And sometimes I also had to work harder on other assignments and give them to my class fellows. So they would give me their bioinformatics assignments and they would help me with the notes. This is how it was. I get started, I come here, it's all about bioinformatics. I was excited I'm going to learn something that's going to teach me about skills that we can currently use in every field. So, and this is how I started. And then I ended up working on two projects. Can we have the next slide? Before that, I would like to tell you that when I got started with my research fellowship, we did a couple of projects that actually helped me understand what the basics of bioinformatics is all about. Then we did the bytes and molecules course, where I also learned about things like metagenomics, and I learned about a little bit about astrobiology, and then the basic tools that we use in bioinformatics, the databases, and a little bit about statistics. Then we came to the transcriptomics course, where I basically got introduced to generation sequencing and how the biological data that is generated from various experiments, or if it's even found in silico, we are able to map the gene expressions and we are able to learn how the gene sequencing is done. And with the help of that data, we can do some statistical analysis. We can look at the pathways that are involved in diseases. We can look at the molecular mechanisms, and we are able to look at the basics of diseases and also with the curative intent and look at some biomarkers. So obviously we've learned all these concepts, we've learned the statistics, we've learned the formulas. Now you also need to put it in the form of a bioinformatics project. So I also did a course where actually I was taught how to write reports from start to finish, what the structure is supposed to be like, how you're supposed to ask your questions, because I think that the question is the crux of your project. If you have a good research question and it's strong enough for you to differentiate your data between the disease state and the non-disease state, or you're able to look at gene expression, which is involves genes that are highly regulated or down-regulated. So we are able to look at the elements that form the molecular basis of diseases, and we can differentiate them. This was the courses that I did. The next slide, please. So my first project was about the transcriptomics analysis of clinical data in COVID-19. The goal of this project was, as we know that last year the pandemic was in full swing and we had several waves. So we looked at patients that were mildly, moderately, and severely infected in various days, which was like the first day. And after a week, the patients were infected with the virus. And we wanted to correlate the severity of the disease with the viral load that they were infected with. And what we did was that we ended up collecting some data from patients, which we were able to find on the Geo website as a database. And then we performed certain tests on it and statistical exams. And then we ended up doing the PCA. So the PCA, it's basically a dimensionality reductional, it's a tool. I know it sounds like a big word right now for people who are like me and just are getting introduced to bioinformatics. But at multiple genes and multiple conditions, we are basically able to look at them in a unified way, that what direction of the disease are they taking us to. And then we also looked at the expression of genes of different kinds. And we were able to see that genes are severely differentially expressed in the mild and progressive stage. So it's also because of how your immune system responds to the disease. And I've noted down some pathways also, like the JAK-STAT pathway and the cytokine receptor interactions, which are able to tell the course of your disease, that if you will be infected mildly or moderately, or it will give you a severe disease. Also, what was very interesting to look at was that when we looked at the viral load, we found no differences between people who were mildly infected and moderately infected and severely infected. We did find differences in their gene expression, like you can see I've drawn the plots also, and we have a bunch of points that are segregated on the plots towards its right and the left, which shows us that there are differences in gene expression. But it doesn't mean that if you have a highly infected viral load and you're just having mild disease, you will not be able to, spread the disease to others. So it's also important for mildly or healthy people to stay indoors and prevent the spread of the disease. Can we have the next slide? Then I did another project over here, which was a part of my master's dissertation. And since I was interested in cancer biology, but I think this field, like since so many people have been working on it, it's become a bit repetitive. So just to make it more interesting or make it more fun, my instructor, Dr. Raghavi, suggested that I could look at the tumor microenvironment. As what the tumor microenvironment does is that it's basically a niche of cells, of growth factors, of signaling elements that our leukemia cells are surrounded with. And we should be able to look at that, is the tumor microenvironment also a cause of being able to generate this disease state, or it's able to turn it's based leukemia, the kind of leukemia that I worked on, it's basically about B cells. So is it able to drive the proliferation of the B cells in such a direction that it becomes disease? And how it exists in certain kinds of tissues like the peripheral blood and the lymph node, as we know that the lymph node is responsible for the growth and for the proliferation of these nascent cells into the adult or full-grown cells, which will then go on and provide immune responses. Again, we did some PCA, we did some statistical analysis like the t-test, so we could differentiate between different groups of the disease. Then we also looked at the differential gene expression, as you can see over here in a figure, that the genes on the right side that are outside of the cutoff and are colored, they were highly upregulated and they were causing certain changes in the cells, such as it could be that the cell's immune function, it could be downregulated, or within the cell cycle, certain checkpoints, if they are released and the cells are and the genes are overactive, it would not be able to cross the checkpoints in the correct pathway, or the growth of the cells would not stop at the required points. That will lead to an indefinite growth and proliferation of the cells. Also, we learned that the tumor microenvironment is able to very significantly affect the progression and occurrence of this disease, as they can as the tumor microenvironment can make the cells highly refractive to the kind of disease that you're in and the drugs that are used for it. And it also increases the inflammation that the cells are increased to, and it's also responsible for DNA damage in the cells. So this is a brief overview, and I'll be showing you the projects and how to write these reports and how to go about the courses in detail. Can we have the next slide, please? So like I said, I have a degree in biotechnology, but sometimes, with such degree, you're also interested in looking at other fields. So I'm currently working as a market research trainee and global data PLC, where I would say that my work is more towards the business side, where we look at the shares and revenues of companies, but we are able to also map it to the concepts of biotechnology that we use in making the medical devices that I've just published. But it would definitely, my dream job would be to again go back to research and look at how these molecular mechanisms are causing diseases and what differentially expressed genes are there, how do we sequence them, and little bit to be able to touch upon the curative approaches. I think this would be the most interesting thing. So I'm going to be working towards that. And coming to the work that I'd done, I had written a poster about it. But if I'm able to gather more data sets which can establish a stronger link between the tumor microenvironment and the occurrence of the disease, or in terms of the occurrence of the COVID disease, and then we draw more diagrams, we make it more systematic, we perform more statistical analysis on it, then I can turn it towards a paper publishing. So now heading to the portal, I'll be showing you my portal, and then you can take a look at the courses, you can take a look at the kind of projects that we've done and how do we submit it.
So we have shared the link to Vedika's profile in the chat so that the participants can go through her profile along the way. So let me pull it up for you.
Yeah, I'm going to share my screen with you so you can take a look.
All right.
Also.
Would you like to start explaining, Vedika?
Yeah, we can do that. Can you go to the courses section?
Yes, we have the courses section right now.
So as you can see on your OmicsLogic profile, you will have a section where you're supposed to talk about your research interests. You can give a little bio about yourself, and then you get started with the basic courses. Like I just said, I was also a beginner advance, and I mean, I barely knew anything about practical bioinformatics except for what I had to do in class to get my grades. Never knew it would look so fascinating to me, and it would just be very interesting. So you can start off with your bytes and molecules course, and then you can also get an introduction to bioinformatics. The introductory course basically tells you what bioinformatics is all about and how you can use big data. Like we know that when we work with such on even in research projects or at a small level, you are required to work with thousands of genes, and you cannot just quantify them at once, or you cannot just look at the differences between them at once. So big data, it's very important to learn those techniques and learn how we can, do the how we can use it in bioinformatics. Then in the bytes and molecules course, you will also get an overview to the kind of fields that you're able to use bioinformatics in, and you will get to learn how to use NCBI. You will get to learn how to visualize genes and how the process of transcription, translation, the biological part of it works. You can do that. And then you'll also learn a little bit about proteins. And interestingly, I learned that it's also important to look at the transcriptome and not just the DNA sequence over here. Shrigori, can you please get to the transcriptomics course? Can you please open it so I can talk alongside? Thank you. So like we say that the DNA expression, it is a bit static since, the genetic expression of cells, you are born with it, but it's the transcripts that undergo more modifications, and they will ultimately lead to the production of the protein products that are responsible for, the occurrence of diseases or the differences in pathways or the botched growth signaling factors, etc. So you will begin with learning NGS, and NGS is also very in technique these days. You will learn how to do the RNA sequencing. You will learn how to prepare your data for downstream analysis. You will learn statistics. And I think the most interesting courses to begin with for me when I learned about RNA-seq techniques, where we were able to look at raw reads and we were able to figure them out, how we map them, how we determine what genes they are. Then coming to the differential gene expression and gene enrichment, which is something that we also do very often in our projects. And then you can also, when you move on, you can go to the machine learning systems, and then you will learn how to do supervised and unsupervised machine learning. And for those of you who are into coding, you can learn Python, you can learn R. These are the languages that basically we use for coding. Since I don't have so much of a coding background, so my instructor, he helped me with the coding part. Sometimes we need to segregate the data. Sometimes we need to look at plenty of data. So I got help with that. And you can also look at other kinds of courses. Like they also have some demo project courses, like the liver cancer one, the one on COVID. Since last year, COVID was in full swing, so everybody wants to learn that. And I think this also ignited our interest in virology again. Otherwise, mostly people are concentrating towards neuroscience and cancer biology. So that is that. You can take a look at the courses of your interest, and you will also learn what databases, what tools to use. And within these courses, what is interesting is that you'll also have some practice exercises. So it's just not reading up stuff on your own in theory. You'll have these practice exercises that you can work on yourself, and then you can come back to your doubts. I also felt like to begin with, when I was beginning with these courses, I think it got a bit difficult for me to understand what these signatures are and how you use these transcriptomic strategies, and how do you look at gene maps, and when you have to combine a lot of approaches, it does get a bit tricky to learn with. So your instructors will help you with that. They'll give you one-on-one classes, and the practice exercises, yeah, like I said, they really help. Now coming to the projects that I worked on, here's another resource that you can look at just for the coronavirus. And you'll also learn about a little bit of the epidemiology. You will learn about how we can backtrace it to the organisms. Like we all know the story of Wuhan and how the coronavirus spread from there. And it's also very related to bats and pangolins, this family of the virus that's infecting people and that caused the epidemic. So we just get to the COVID-19 project, and I'll take you through it. So while you write your projects, you have it's a set of steps that we have to follow, and I think it's something that we do everywhere. Even when we write a report or you want to do a research paper, you will start off with collecting a data set, then working on it through your materials and method section, and then you should be able to derive some significant results, and you will be able to look at the data that way. So then you write an abstract about what you do in the project. Then you give an introduction to the theoretical part, like you could talk about what the coronavirus is all about, what are the stages of the disease, and how do people get infected. Then you will tell them the methods that you did, like I just talked about the statistical analysis and the principal, the PCA and stuff like that. Then you can discuss what did you learn from the project. You can also talk about your results before that. Obviously you'll be doing the results first and then the discussion. What is it that you can draw, and what can you attribute the diseases to? And it's not only limited to diseases. If you're interested in the agriculture side, or if you're interested in the microbial side, you can also do projects like that. Then you'll conclude your project, you'll give your references, and then you'll give an acknowledgement. So this is a format, this is like a standard format that you should keep in mind when you are writing a project. So basically, when I did the COVID project, what we did was that towards an introduction, we were able to look at the genome of the COVID virus. A little bit, we got an introduction to it, that it has about, you can say it shares about 80 to 90% of its sequence with bat and pangolin virus. And then the gene, or I would rather call it the DNA of the virus, the genetic material or the RNA of the virus, it codes for various segments of the virus, like the ORFs, and then the envelope and the membrane and the spike, and then the capsid protein code, like we all read about in our virology classes. And sometimes they also have some accessory proteins that help the virus penetrate better into the cells and cause the disease. So this is some basic introduction to COVID. And then what the spike protein of the COVID does is that it binds to ACE receptors, which are also some receptors that are like highly concentrated in our heart and lungs, and many drugs are also made against these receptors. Then the COVID virus from your nasal tract, it goes into your lungs, and then whatever the disease severity has to occur, it occurs. So like we collected this data for this project, and we collected about 180 samples from 98 people, which was segregated into the base day of the disease, the day they were diagnosed, and then a week later or 5 days later, where you can look at the progression. Then we did the PCA for it, where we kept a threshold that if the gene expression it occurs beyond a certain level, then only we will consider it significant, which was done in the quantile normalization step, where basically you will transform the genes into a bunch which can be compared with each other, because you don't want any kind of background noise or genes that are not significant or very mildly expressed that won't change the course of the disease to affect your results. Then you do some differential gene expression about it. You will select thousands of genes. You will cut them down to the genes that cause the disease. And then you can help with the help of various diagrams and charts and bar plots, you can look at the results. So we found out that, like I've written here, that the COVID disease, it gives you a very hyperinflammatory state. And there are various pathways and genes that, will also affect the ability of the immune responses that that the host will do to the viral proteins in the form of cytokines, etc. So this was a little technical sci-fi facts of the project. And then I'll also walk you through the other project which I had done as part of my final year's master's dissertation. I came back here again because like it was COVID and what would you think of doing? So, the like all these institutes and stuff were closed, but I'm so glad I did it. It was a very interesting project to work on. And the good thing about it is it's all of it is so interactive that you're able to visualize it with your eyes, you're able to dive into genes, and you're able to look at the results instead of just sitting in class and just reading theoretically about things which I know can get a bit dry and uninteresting to work on. So to begin with, for a project, you will also get some data sets, which you will, like I said, you can obtain it from the Geo website or any other database portal related to your disease. And then you'll get the counts of the kind of genes and the gene expression that occurs in a disease. You can take the raw data and then you can work on it. You can perform your statistical tests. You can annotate it to the various gene IDs. You can look at the pathways that are involved in the disease. So we'll just get to the introduction and the method section. I can just talk about, so you'll basically again get an idea about what the projects are all about. So again, chronic lymphocytic leukemia, it is a disease that affects the B cells, where the immature B cells, they undergo such high levels of proliferation that the count crosses a certain value, which is obviously medically kept as a cutoff. And it can cause, that's why it can cause various symptoms and the stages of the disease. So what we did was that we collected some data from Geo. This time, like I said, that, not only the disease, you can also look at the various types of tissues that are involved in a disease. Like we did peripheral blood and lymph node. And we also looked at other conditions of the kind of cells and the markers that they showed. We chose CD19 because it's highly, I would say, like, a statutory when it comes to this disease. So it helps us differentiate between healthy and the disease cells. Then we again did the PCA, we did some differential gene expression. And a lot of these steps were done on the T-BioInfo Server's platform, which is here you can look at the pipelines, you put the data in and you get the results out with the certain conditions that you expose your data to. Then we had to also statistically determine that if the genes are differentially expressed, do they even make any sense for them to be this way or not? So we did that, we used certain formulas and tests, and then again we did the differential gene expression. And with the help of gene ontology, we were able to concentrate the genes that affect the molecular and biological functions of the disease. So you can use various online tools. You can also code and look at the kind of diseases and the genes you're looking at. So that is one thing. If we get to the results of the project, you can just see that it looks very interactive when you do it in the form of charts and plots, and it also gives you a lot of clarity. So like you can see that the various subgroups of the disease, like lymph node or blood, or the disease, or the cells that were healthy enough, and the unhealthy cells that also had markers, we determine them, their propensity to cause a disease in the terms of certain percentages. And you see that how they're differentiated on the charts. Also, you can, like I just talked about in my previous project also, that the upregulated and downregulated genes, we can see not only how much they change and how they change and what these mutations are able to cause. Now coming to the discussion and conclusion of the project. So we did learn that various signaling pathways, basically, there was a very interesting pathway called the notch pathway that is responsible for causing changes in the tumor microenvironment, which is able to cause the occurrence of this disease. And the notch pathway, along with some other signaling pathways, that we usually read in our cell biology classes, they will also affect the tumor microenvironment, which can interact with the cells and determine the fate of the cells if it grows into a network of healthy cells or unhealthy cells. Also, we can note certain, like I've given the names of certain genes, like it is the PIK3CD gene. It is involved in the proliferation of the cell, it is involved in the development of the cell, and then T cell receptor signaling. Like it tells us that, it's not just limited to the B cell proliferation, but the other kinds of immune cells also, they all work in a niche, which is where the microenvironment comes in. Then certain genes that will affect the metabolism of proteins and lipids, which can lead to the production of certain signaling factors, which may be faulty or which may be healthy. And there are also genes which can mutate. It can contribute to the drug resistance or the reoccurrence of the disease, which is also there. And then, since we know that most of these mechanisms are molecular and they involve the DNA sequences and stuff, so you will also look at CLL patients, they will have higher rates of translation, and the regulation by the ribosomes of the genes, they will be lowered. So that can also lead to the occurrence of the disease, as these signaling, when they occur in the DNA and RNA, it will lead to the occurrence of certain kinds of faulty cell cycle, and the mitotic spindles may be affected. And these are just like a bunch of phenomenon that can occur with the occurrence of this disease. What it can also help us do is, it can look at we can look at biomarkers, that if we see that there are certain genes or that are differentially expressed, and they can affect certain functions of the cells, like the DNA repair will be altered, the signaling pathways, they will be altered, it can cause an inflammation, or it can affect the organelles of the cell, like it may cause some mitochondrial dysfunction in the cells, and it can also cause some faulty occurrence of the cellular processes and signaling cycles, like we read about the calcium dependent kinases and the BCR receptors, etc. So this was about it. And when you do it in silico, it also gives you a very thorough look at what, like I worked on the disease side of things. So I think it is a very thorough and a very interactive way to look at things. So I'm very happy, and I'm actually very happy to be coming back here today to talk about my projects and my journey as a research fellow. And it was very interesting, and I'm so thankful that I was able to do this side of bioinformatics things. And I'm always looking forward to learning more about such stuff. So thank you for this. And if there are any questions that you would like to ask about writing a project or anything just in general, please feel free to ask.
Yes, we do have a query from Uzma Manzoor, and she's asking, have you done all these courses online?
Yes, Uzma. So like the fellowship that we did, we mostly do it online, and you can see that they have a portal of the courses. So what you're supposed to do is, you're supposed to go over the study material. They will give you certain videos also in the study material. Like when I started with the transcriptomics course, I remember I was able to look at how the next generation sequencing is done, how the I would say the samples are prepared, how do we map them to the genes, and what strategies are used. So you should be able to, mostly you will be doing these courses online, and then you can always get in touch with your instructors if you have any doubts, or if you are stuck somewhere in doing the assignments. So that is how it works. And they've divided into lessons for each topic. So I think it makes it easier to understand things, and you can go at your pace, and you can start off with the easier things and then work your way up. That is how we do it.
Thank you so much, Vedika. And I hope your query got answered, Uzma.